Hello. We know that cells replicate, and we know that each of the cells have genome. That means there should be some coordination between the cell division and the replication. And how is this accomplished is a complex process. And if we are trying to read or try to understand both that of prokaryotes and eukaryotes, then it's going to be a little bit, there will be large number of variations as well. I am Dr. BCMR, Assistant Professor at Shastra. So majority of the prokaryotes, they have circular DNA. And eukaryotes, most of them have numerous and they are mostly linear DNA. And there is a complexity then that when should replication start, and by what stage of cell should replication finish. For bacteria, it is much more relaxed in that sense compared to that of eukaryotes, because bacteria have cell division, and one bacterium undergoes mitosis and produces two. If we talk about something, an enzyme, a bacterium like that of E. coli, and in the best conditions or optimal conditions, they can divide every 20 minutes, and that is one of the fastest growth rates an organism can have. And every cell, there is a genome, and replication of that genome requires certain amount of matter, and it also takes certain amount of time. And at the same time, the cells, the remaining cytoplasm, cell organelles, if there are any other, and other components, they should be doubled, and only then the cell division can occur and produce two viable cells. The objective of every cell division is to produce, or majority of the cell divisions is to produce viable cells. And in that way, bacterial cells have the nucleoid, what we call it as, is because they do not have a nucleus. The genome itself, along with the proteins associated, we just refer to it as the nucleoid. They have a region, that is the replicator, a DNA molecule with an origin of replication. We can call it as a replicator. So there is a replicator sequence to which an initiator binds, and then it starts it will allow the loading of helicases, and then helicase loader will also be there, of course, and there will be several other proteins too. And the helicases will start unwinding, and the two replication forks will keep moving the opposite directions. So we have replication bubble, and that matures into two replication forks moving in the opposite directions like this. And eventually we will have two interlinked double-stranded DNA molecules, which will be resolved by topoisomerases. In eukaryotes, we have numerous DNA molecules, and they are very large. So because they are very large, and then they all have to be regulated and finished by a certain period of time, each of the chromosomes have multiple origin of replications. You can also, we may have this autonomous replicating system. That is another word almost used synonymous to that of origins of replication. So the chromosomes have multiple of them, and at that place the initiator protein binds, and then the loading of helicase and other proteins will happen, and then the replication fork will move opposite directions, and eventually we will have two DNA molecules produced like this. That is the general overview of how replication is coordinated or happens to start with the initiation of replication. If we look at a little bit closer and try to understand molecular mechanisms, the origin of replications have certain regions like this. This is a 9-mer, meaning that there are 9 bases here, and we have repetitions of these 9. Of course, in different organisms there are different sequences, and number of these mers will also be different. And the origin of replication in E. coli has 13 mers, 3 of them, and 9 mers, there are 4 of them. and there is a protein that binds to that. That is, the replicator is this 13 mers plus 9 mers, and the initiator protein here is DNA A, which binds to ATP and in its inactive form, and then it binds to the 9 mers, and because of this binding there is a torsional stress, or so, because of which the 13 mers region will open up something like this. And 13 mers are rich in AT regions, which would mean also that there are less number of hydrogen bonds, and then the opening or the initiation or the formation of replication bubble is much easier. And once that happens, there are several proteins that bind to it, as shown here. We have the helicase loader, which will also load the helicase, and helicase will, once it binds, and they will keep on moving to the opposite direction, and then the organization of the replication fork. So we do need, once the with helicase there is also primase that is associated, and then we have the recruitment of DNA polymerase 3 holoenzyme along with sliding clamp and sliding clamp loader. So the whole replizome is organized in that location, and then replication continues. So there are some mechanisms to control when the cell division should happen. Say for example, if we think of it, say this is a DNA replication, so we'll take it as circular DNA, and then we will assume that the replication bubble has opened up. So we would have new DNA molecules. Assume this is where the origin of replication is. So now we have DNA that is newly formed here. This is the DNA that is newly formed. Here is a replication fork moving in this direction, and the other one moving in the opposite direction. So now we have the origin of replication here. So should this reinitiate initiate replication or not? That is the question. Or should it wait until one round of replication is completed and then resume the second cycle of cell division, or replication. So E. coli has, or every organism has, mechanisms to regulate that, when and then how to prevent the immediate initiation of this replication. So that is by a mechanism that we call the initiate region, or the origin of replication region, ORIC, has several GATC sequences like this. And there is an enzyme called as DAM methylase, which methylates the adenine, N6th position of the adenine in all of these. So now we have a methylated DNA, and methylated in these GATC regions. And once replication happens, then there is something happening. One of it, because there is semi-conservative replication, one of it will retain the methylation as is here, whereas the new strands will not have any methylation. Right, the same GATC sequences I am indicating here as this as methylated group, the adenine within the GATC. So if we see in this region, we do not have the GATC regions in that location do not have methylation. So those are referred to as hemimethylated. You can think of, say, semi-methylated and hemimethylated, right? Here at least hemi is about all the methylation, if there is only present on one of the strands, and the other strand remains unmethylated. Those are called as hemimethylated. Say for example, we have several regions like these GATC regions, and we have methylation in some locations but not in one strand like this and in the other like this. That probably is semi-methylated or partially methylated region. So at least the term hemimethylated, one should remember it as one strand is methylated whereas the other strand is not methylated or is in unmethylated state. So what happens in that case is there is a protein called Sec-A. Sec-A is specific to hemimethylated regions. So if GATC hemimethylated regions are there, Sec-A protein comes and it binds here. Because the origin of replication is now occupied by Sec-A, a protein, DNA-A cannot bind. DNA-A, if you remember, is the initiator protein. So because this region is occupied by Sec-A, it cannot be occupied, or this part of the DNA cannot be interacted upon by DNA-A. Therefore, replication initiation, the second round, is delayed. But the Sec-A binding to this region is not permanent. It keeps dissociating now and then. Every time there is a dissociation, so we will retain it, we will keep, now that one of the replication is completed, we can assume that. So this is the, I am only showing one of the two DNA molecules. Sometimes Sec-A, because it doesn't bind, at that time DAM-methylase can come and methylate this region as well. So it takes a while before this region gets methylated. All the GATC regions within this, the origin of replication. And once they are completely methylated, Sec-A can no longer bind there, and then DNA-A can bind and reinitiate the replication. There are several other based on DNA-A and ATP ratio 2, because DNA-A binds to ATP and only then it is active. If the cellular energy levels are less, then the ATP to ADP ratio is less, meaning that if the energy is not in abundance, ATP levels are less and relatively ADP ratios are higher. In that case, DNA-A is not in active state, and then therefore it cannot initiate replication. That way, when conditions are not very optimal, the replication can be delayed or actually halted. Initiation of replication. We all have to remember that regulation at the beginning, that is the initiation, is everything when it comes to DNA. We cannot regulate it in the middle of elongation. There is no point of it. It is more vulnerable to damage. So whatever it is, it has to do it during the initiation. The same way we have also with eukaryotes. Eukaryotes have much more complex. They have cell organelles, cell division is much more orchestrated, and there are eukaryotic initiation is much more complex and highly interwoven with the cell division. So here the origin sequence, and what we call it as origin recognition complex, it is a multi-protein complex, and it is interacted upon by CDC6, and then there are many things that happen, which include also the loading of MCM, which is nothing but the helicase, and the helicase will bind and will start unwinding it. There are many other proteins involved in it. We are not discussing in that great detail, but you can refer to the textbook. But I still want to take you along with how it is coordinated with the cell division. We will start off with G1 phase. I hope we can recollect G1 is gap 1 phase. S is for synthesis, that is where replication actually happens. And then we have G2 phase, second gap phase, where the cell is preparing for mitosis. And then in M phase, it's either mitosis or meiosis. How is initiation regulated, or what is the status of the origin of replication within these different stages? We'll go here in the first gap 1 phase. Helicase is loaded, that means, but helicase is loaded. So ORC binds CDC6 and helicase is loaded, but helicase activity is prevented. There is no helicase activity because CDK is low. we should understand that cyclin-dependent kinases, there are a lot of phosphorylases, phosphatases involved, that these post-translational modifications can control. In the S phase, we have the helicase activation, but no more helicase loading, and that is because of CDK levels are high, because of this phosphorylation of several proteins that are recruited to the origin of replication. Helicase activation happens, but no further helicase loading can happen. In G2 phase, origin recognition complex can be loaded, and there is no helicase activity because there is no helicase loading to that. And in M phase, whatever origin of replication loading has happened in G2 phase will remain as such. There is no helicase loading, so no points in it. So in G1 phase, that is where helicase loading happens, but all the origin of replications will have helicases loaded onto them, but there is no helicase activity. Only when it enters from G1 to S phase that the helicase activation or the helicase activity is initiated, and then the replication of DNA is taken place. I hope we have a pretty fair idea about how regulation happens. Regulation of initiation of replication happens in prokaryotes and in eukaryotes, at least briefly. I would suggest always to read the textbook, which will give a good idea. The majority of the material that I am discussing is from the Molecular Biology of Gene by James Watson and other co-authors. Thank you very much. Have fun.